A scientist is studying an unknown plant species with two flower colors, red and yellow. She finds a rare red flower plant in the wild and brings it to her lab to breed. She crosses it with another plant and gets two different types of offsprings, 50% red and 50% yellow flowered offsprings. The scientist is puzzled. She expected to only find red flowered offsprings, but the appearance of yellow flower means she miscalculated something. So on to our question. We have to find out the genotype of the original red parent. The original red flower plant can be determined as R dash, and there's two possibilities. It could either be capital R or capital R small r. Now, to figure out the genotype of the original red flower plant, we have to test cross our unknown organism with a homozygous recessive plant for the test cross. So our options are capital R with a small r, or a capital R small r with a small r. Trying our first option, we will have to draw a Punnett square. So our plants are, the offspring plants are capital R small r, capital R small r, capital R small r, capital R small r, which is 100% red offsprings. And for the second option, we have capital R small r crossed with small r. So we have capital R small r, capital R small r, small r. So this is 50% red and 50% yellow. Since 50% of the offsprings have to be yellow flowered plants, that means the original red flowered plant passed on a recessive R allele, so it must be capital R small r. This is the only option that is consistent with our result. So this is the genotype of the original flowered plant. Capital R small r. Now on to our next question. What was the genotype of the other parent in the cross? The other parent in the cross has to be a small r. What is the dominant and recessive flower color? The dominant color is red, capital R, and the recessive color is yellow, small r.
In fruit flies, body color and wing shape are controlled by two genes. A fly with gray body, big G, dominant over black, little g, and normal wings, big N, dominant over vestigial, little n, is crossed with homozygous recessive fly, little g, little n. And the four phenotypes we got were 420 gray normal, 410 black vestigial, 85 black normal, 85 black normal. Question 8 is asking, based on the observed offspring, determine whether these two genes assort independently or are linked. And to figure that out, we just need to know if the genes assort. If the genes assorted independently, we would expect a 1:1:1:1 ratio of the four phenotypes. Since the observed offspring do not fit a 1:1:1:1 ratio, the genes do not assort independently and are instead linked. Question B asks, if they are linked, estimate the recombinance frequency. The RF formula would be used for this. And what we do is we take the number of recombinant offspring divided by total offspring times 100. And our recombinant offspring equals 170, and our total offspring equals 1000. We take 170 over 1000 times 100, it equals 17%. Since recombination is less likely than 50%, the genes are linked. The RF of 17% means the genes are 17 map units apart on the chromosome.